Hi, Lee.
Hi, Carl.
Let me introduce myself first. My name's Carl Zimmer, and I'm a science writer. And Lee, could you introduce yourself?
Sure. I am a professor at Princeton University in the departments of molecular biology and the Woodrow Wilson School of Public and International Affairs.
So, one reason, Lee, that I wanted to talk to you was that I really enjoyed a book of yours called Challenging Nature. I was reading it a few months ago for a book that I'm working on about E. coli, actually.
Well, thank you. Appreciate that.
Well, I was getting interested as I was working on E. coli and these questions of sort of what's natural and what's moral when it comes to science, because E. coli kind of, in a way, sort of set off this debate with genetic engineering, and now, 30, 40 years later, we're dealing with issues like stem cells and genetically modified foods and things like that.
And even beyond that, Craig Venter and others are trying to create completely synthetic organisms, so that's a step beyond recombinant DNA and genetic engineering.
Yeah, so I liked how you really sort of we're kind of putting the issues in pretty, stark terms about how we deal with these things and the values that we bring to them. And then when there was all this stem cell news that came up recently, I thought, wow, it would be really interesting to talk to Lee about, what's been happening and what you think about it. So
sounds good. So go ahead, fire away, and I'm ready to discuss.
Great. Well, maybe we should describe a bit about what this recent news actually has been, because there's a lot of sometimes there's more heat than light in the coverage of these things. So,
right.
I mean, the ongoing controversy over stem cells has been over deriving them from embryos, and then recently there was this advance where it was actually possible to derive stem cells from skin cells. I mean, can you go into a little more detail about what exactly they did?
I think it's worthwhile sort of giving a summary of both the scientific and the political portion of the issue. So originally, Jamie Thomson did this with human cells, but actually it was done back in 1981 by Gail Martin and Martin Evans on mouse cells. And what they were able to do is to take early embryos, pre-implantation embryos, a couple of days after fertilization, and out of those embryos they were able to grow what we call embryonic stem cells. Now, as I said, this wasn't done with human embryos until 1998 when Jamie Thomson reported it. But the political contention is actually not over growing embryonic stem cells per se in a petri dish, but over this notion that you have to destroy a human embryo to get the embryonic stem cell. So that's what the contention has been over the last 6-7 years. Mm-hmm. Is the question of, is it moral to destroy? I have a problem with the word destroy, but that's the way people refer to it. Is it moral to destroy embryos to make embryonic stem cells? Right, sure. And now what has happened just recently, reported both from Jamie Thomson's lab and from the Japanese group, is taking adult cells, they're referred to in the press as skin cells, scientists call them fibroblasts, and
but they're still in the skin, but they're not like epidermal cells. Is that
right? They're not on the surface of the skin. I mean, it's easy to call them skin cells because you can get them from anywhere in the body, basically.
Oh, I see.
And these are fibroblasts. Fibroblasts are very easy to grow in the laboratory as well, and they figured out what gene products were necessary to convert these skin cells into embryonic cells. And these two groups actually used they both used 4 genes. 2 of the genes were the same with each of the groups, and 2 of the genes were different. And essentially what they did is they actually added the genes themselves to the cells. These genes created the proteins that they code for, and then these proteins essentially rebooted the skin cells into an embryonic state.
Mm-hmm. Meaning what? That they
we'll talk more about that, but meaning essentially that if you looked at these cells after this transformation, these cells now had properties which have been attributed to embryonic stem cells, both molecular properties as well as differentiation properties. In other words, these cells, if you look at their molecular constitution, they looked like embryonic stem cells. And then if you put them into different environments, you could actually get them to differentiate into the various kinds of adult cells that exist in the body. Mm-hmm. So that basically, that's it. I mean, that was the report, or actually the two reports. And I actually think it's a very significant finding, not just for the science, but also for the philosophical implications.
Well, let's take the science first. I mean,
Sure.
Just setting aside all the politics and so on, I think it's pretty remarkable that you can take a cell that has become so specialized in an adult and with four proteins, just take it all the way back to this very different state. I mean, does that tell you something about development itself?
Well, this is not unexpected from
really?
I would say from those in the field. No, I think the Dolly announcement was the giant leap forward, and so essentially what Ian Wilmot and Keith Campbell did with the Dolly experiment is they took an adult cell. It was a mammary cell, but it could have been a fibroblast, and they took the DNA from that cell and stuck it into an egg whose own DNA had been removed. And the proteins or factors in the cytoplasm of the egg did the same kind of thing. It transformed the genetic program that was being run by the DNA. And so that's how they originally were able to clone Dolly. They took an adult cell, they took the DNA out of the adult cell essentially, and they reprogrammed it with proteins that existed in the egg cytoplasm. So in my mind, it was just a matter of time before scientists discovered what exactly the proteins were that were sitting in the egg cytoplasm that reprogrammed the adult genome. Right. And that's what this new discovery has done. It's revealed to us a set of specific proteins that allow this reprogramming to occur.
I see.
Yes.
And so there's the science, and then there's the philosophy. You said. I mean, what?
Yes. I mean, the biggest shock about Dolly was that this dogma, this biological dogma that existed for 100 years was shattered. I mean, almost every scientist, thought that once a cell had differentiated and had become an adult cell, it could not go back. Now, there actually was never any strong, at least recently, there hasn't been any strong reason to believe that, because the adult cell, nearly all adult cells have the same DNA constitution as an embryonic cell, and it's the genes that are being used to create the cell's components and determine the cell type. So an adult cell has the same DNA, the same genes as an embryonic cell, and the difference really is how those genes are being used, which portion of the total gene complement is being used, and at what levels are the different genes being used to create proteins.
So it's sort of like all the same instruments in an orchestra, they're just playing differently.
Yes, they're playing differently. I mean, the other thing is just to consider it like a Windows computer is, you get very, very, I don't use Windows computers, but I know those who do, you get very, very far along, something goes wrong, and then you do Ctrl-alt-delete to get back to the beginning, and the software's still there. So the question is, can you take an adult cell and do Ctrl-alt-delete and bring it back to the embryonic state? And Dolly showed you could. But Dolly was a really brute force method, because you had no idea what the proteins were. You had a very complex mixture of proteins in the egg, and clearly some of them, and maybe some non-proteins as well, were going into the nucleus, the adult nucleus, and reprogramming the genome.
Right.
And so what this recent result shows us is the precise proteins that can be used to accomplish this task.
Mm-hmm. So you don't just have to, so you don't just have to just come in there and with the brute force that they were using, you can
Right.
be more precise about it. I mean, do you think that maybe that would be able to help in terms of avoiding some of the problems that people see about using stem cells, about
So
how
you're
they
talking
might
about the political...
differentiation? Well, I was just thinking about sort of like the possibility that a stem cell could become cancerous, for example. I'm just wondering if you have sort of finer control over them.
Yes, I mean, that was always a technical problem with stem cells, is they can become anything, including cancerous. But I think that's a technical detail. I mean, before any of these before this technology is going to actually be applied in a real therapy, you have to work that out. You're going to have to make sure that you're probably not going to put the stem cells in. You're going to put the cells that have differentiated out of the stem cells in your petri dish back into a patient. And you're going to be doing it under conditions where you've demonstrated with animal models that it's not going to cause cancer. So I think this is an incredible step forward for many, many reasons. And the first step is that you don't need human eggs to make embryonic stem cells, assuming that this research can be reproduced, and I have every reason to believe that it can. So you eliminate the whole ethical problem, which everybody accepted is an ethical problem, of having to pay women to take their eggs to use in this technology. You can eliminate that problem completely. It's a much easier experiment. I mean, it actually takes a lot of skill to manipulate eggs and to inject nuclei or cells into eggs. You don't have to have that skill anymore. And the third point is, again, assuming that this can be reproduced, it's very straightforward, and it's not going to be contentious. So it completely turns the political scene concerning stem cells upside down, again, if this can be reproduced. So there's no reason not to do it this way. I mean, it's not just stem cells that you're creating. You're actually creating cloned stem cells from the individual that gives the adult cell. Of course, interestingly, the scientists who have done this are avoiding words like clone and embryonic stem cell. Well, they're going to call it something different.
Well, I mean, there's been some talk about how some people just want to try to stop all the embryonic stem cell work because now we've got this alternative. I wonder if they'd actually be able to do that, first of all, and if they did, I wonder if stem cell research would really be ready yet to lose all of that side of their research.
Well, I think, and I'm somebody who's always been very supportive of embryonic stem cell research, but I think the game has changed, and actually a lot of the supporters don't realize how thoroughly the game may have changed right now. I mean, if these new cells, which have been given a new name, they're not called embryonic stem cells, they're being called induced pluripotent stem cells, or IPS cells. So, and that's playing games with words, but it's playing games with words to the effect that people are not scared by this, and nobody is going to be opposed to it. These cells are supposed to have the same properties as embryonic stem cells have. And if that is true, and other scientists can reproduce this research easily, then this is the way you're going to get embryonic stem cells, which are now not called embryonic stem cells. So it completely changes the game, and I think a lot of the supporters of embryonic stem cell research are being extremely cautious. They say, well, we can't stop working with embryos yet, and I would agree until this has been replicated multiple times. that's true. But I think ultimately, there's not going to be any reason to go back to eggs, actually, to do this research.
Mm-hmm.
So, and that has, it has huge consequences for the countries that have poured a lot of money into this, the states that have poured money into it, because all of a sudden it's going to be a level playing field in terms of where this research can be done.
So that you don't think states like California might have gotten kind of a head start because they had already been working with the embryonic stem cells?
Oh, I think they do have a head start. Yes, it's a head start because they already have funding for the research. They've already attracted a lot of scientists from across the country to California. And even before the embryonic stem cell initiative in California, California has always been an attractive place for biomedical scientists to go. There's a lot of biomedical scientists there. There's a lot of support for it. So in that sense, they have a head start. But politically, the field is going to be leveled. I mean, it's still going to be the case that even though nobody's going to have a problem with this research in Idaho, as an example, I don't think you're going to have a lot of scientists running to Idaho because they don't have the infrastructure to support it.
One thing that I liked in your book, Challenging Nature, is that you really tried to get down to the sort of the core of these ethical arguments over things like stem cells,
yes.
Which I find that are often people talk about these things very passionately and have very deeply held views, but it's hard to kind of pry out sort of the logic that people have about why something is, say, unnatural or even immoral. And these stem cells are so interesting because, in a sense, well, the way that I've seen this development spun in the press is that this completely gets us around this ethical problem that embryonic stem cells pose. But really, as in a sense, you don't, because these new cells they have a lot of the same properties as embryonic stem cells, and in a sense, they could become embryos. They could become life. How do you see the?
Well, you see, this is a game, and the first question is, how do you look at cells? I mean, are cells just complex information processing machines, which is the way most biochemists and molecular biologists look at cells? Or is there something mystical and vitalistic about certain cells, embryo cells, for example, as compared to other kinds of human cells? And the opponents of embryonic stem cell research base their opposition on the belief that there is something fundamentally different about one kind of cell, the cell that is an embryo, one cell embryo, two cell embryos, because each of the cells in a 2-4 cell embryo has the potential to create an entire human being. We know that from identical twins, identical triplets. So is there something fundamentally, ontologically different about embryo cells, human embryo cells, versus human skin cells? And some people think there is. Now, as a biochemist, molecular biologist looking at it, the only fundamental difference is there are different proteins sitting on the DNA in different places, which causes these two cells, which have the same DNA, to look a bit different from each other, to have different potential relative to each other. That's not a fundamental difference. And in fact, this new research has shown how unfundamental the difference is, that you can go back and forth. But the public has this real sense that skin cells are worthless, embryo cells are sacred. And so this new research has produced papers, the paper from Jamie Thompson's lab, where the scientists are very careful about the language. So this is political science. This is not molecular science. And the political science is, we're not going to call them embryonic stem cells, even though the whole point is that they are embryonic stem cells. And we're not going to call them clones, even though, in fact, they are clones. we're going to avoid those words. Those words are contentious, and by avoiding those words, everybody's going to be happy. And that's basically what happened here. And I think it's going to work.
So do you think that that people are just going to be able to kind of overlook these kind of contradictions, and we'll all just sort of go ahead with the stem cell work using skin cells and not look in the direction of these ethical issues anymore? Or do you think that we're going to be running into them again?
No, I think I've talked to many opponents of this research. The Catholic Church is very opposed to the use of eggs or embryos to create embryonic stem cells. There are also the opponents are all Jewish or mostly Christian, and they have this view that embryos are different from other kinds of cells. And very often they won't admit it, but it's a vitalistic notion.
Vitalistic meaning what?
Meaning there's something beyond the material substance of an embryo that gives it its properties, that allows it to develop into a human baby. And those properties, those beyond materialistic properties, don't exist in the skin cell or in and what the cell's being called now that is embryonic stem cell like. It's being called induced pluripotent stem cell. That's a real fudge, because if these are really embryonic stem cells, and they certainly look like them to me based on the data that I've seen, we know that the mouse version of these cells can produce a mouse. And there's every reason to believe that the human version of these cells can produce a human being, but the scientists are going to make sure we don't talk about that. And I think that the opponents, they're pretty, it's very confusing when you start talking about these cells. There's this notion of the absolute difference between cells, and this notion is false. I mean, all the cells have human DNA, and they're just expressing somewhat different programs. That's how I would see it as a molecular biologist. But the view of opponents of the original research was that there is this qualitative difference. And that's why induced pluripotent stem cells sounds good. It's not something that's going to upset people.
I see.
So I think it's going to, that's why I think this is going to revolutionize the debate. I mean, it's an interesting scientific advance. I had no doubt this would happen. As usual, I thought it would happen in 5 or 10 years, and it's happened a lot faster. But I don't think people realize yet that this is going to completely turn the political background of stem cell research upside down.
Well, when you say it's going to turn upside down, do you mean it's just going to? I mean, is the debate just going to end?
Pretty much. I think it's going to end. I really do. Wow. Now, I may be wrong, but I just don't. I think that if this work is reproduced and the data look very good, and these are two laboratories that are very reputable, and they've published all the details, it means other scientists should be able to reproduce this this result in a matter of months.
Yeah.
And if they can, I think the entire political issue just is going to disappear.
Or maybe it's just going to move eventually to another application. It would be one thing to be using stem cells it would be one thing to take these skin cells and make these induced pluripotent
iPS cells.
Right.
Right.
I'll get used to this term. I'll be able to roll off my tongue eventually, but. Anyway, so you take these cells and you put them in a lab and you use them to study diseases in a dish. I think that will just sort of move along quietly the way so much biomedical research does. But then, I mean, somebody's going to take some of these cells probably and stick them in a mouse's brain or something like that, right?
Oh, well, yeah, and that's going to be.
And then we're going to be hitting these same issues all over again.
Well, that portion of the research agenda is contentious. Yes, that's always going to be controversial. The portion that I don't think is going to be controversial is, okay, we're going to take these cells, they're adult-derived. They're not embryonic derived. That's the whole big thing here. You're going to be able to study diseases in the laboratory, and you're also going to be able to figure out how to take these cells and turn them into blood cells and turn them into liver cells and all the various other kinds of cells that you might need to overcome disease. And that's not going to be contentious. You take somebody's, you're taking a patient's skin cell, making this so-called iPS cell in a petri dish, sprinkling some proteins on it so it turns into blood, or hematopoietic stem cells, really, and then putting those back into the patient. I just don't see that as being controversial. It's going to be like every other biomedical procedure. Now, you did bring up, of course, the controversial portion of this research, which is, okay, we want to study neurological diseases, so it would be good to put these cells into a mouse brain and have a mouse whose brain is part mouse, part human. Yes, that's still going to be controversial.
It would be, I don't know, it's interesting the way you talk about it. I mean, it maybe 10 years from now, people are going to look back at, the stem cell controversy years and just sort of scratch their head over it. I mean, if it's really...
Well, it's happened. It's happened before. I mean, in two different examples are worth considering. When the technique nuclear magnetic resonance from physical chemistry was first used in a clinical setting, patients were afraid of it because it had the word nuclear in it, and so the technique in the clinical setting was renamed magnetic resonance imaging, MRI, and now everybody's happy with it. And that's word games. I mean, everybody accepted it because those in the know knew that the nuclear part of NMR has nothing to do with radiation. Yeah. So, everybody sort of played this game, and everybody's happier for it. So that's one example where a change took place. I think the other example is that the original recombinant DNA technology of the 1970s...
Right, I was thinking about that. I mean, I've been looking a lot into that, and I think I agree it's totally the same situation.
Yes, I mean, I remember being a graduate student at Harvard when this was... the controversy was ongoing, and
we should just describe what was happening. So in the 1970s, people were figuring out how to take genes from other species and stick them into E. coli and get it to make the proteins from those genes.
Right, and the most evocative example was to take the human insulin gene and stick it in E. coli and get E. coli that produce insulin protein that can be used to overcome diabetes. And that was extremely controversial in the 1970s, and they tried to ban it in Cambridge, Massachusetts, where a lot of the research was going on. Now, opponents of biotechnology come out explicitly and say they don't have a problem with that. They've changed the tack, and they say, of course, that's a very good use of biotechnology, but now we don't like the new kind of technology that manipulates human cells, or especially genetically modified crops. They don't like. But everybody's come to accept the manipulation of E. coli to make human pharmaceuticals.
Right. I mean, it's a billions of dollars of insulin.
And there's lots of there are a lot of diabetics, and everybody has a family member who's diabetic. So, of course, I mean, there are a few holdouts still, but this is different because I think here, scientists used the wrong language initially. They didn't realize. Now they're using a different language. So embryonic stem cells, I think, are just going to disappear from the jargon. I mean, nobody's going to use that terminology anymore. So, there are a lot of people out there, my friends, who say, well, we have to be cautious. Maybe there's a reason to keep on thinking we're going to need embryonic stem cells. But when I think about this deeply, I just think it's just not going to happen. This technique is easier. It's not contentious. It does everything you want it to do. Again, assuming that it can be replicated.
Well, I mean, that brings up another question, which is that sort of going underneath the surface in the stem cell debate has been, the question of how useful these things really are going to turn out to be. I mean,
right.
And opponents of what I will call embryonic stem cells would say that they would claim that there was a lot of hype being generated to justify the research on these particular kinds of stem cells, and that they were raising false hopes and so on and so forth.
Right.
I mean, I find it interesting actually that everybody is now looking at these skin cells and saying, "Oh, it's going to be great. It's game over. This is solved all the problems." So the same. Sometimes it seems like the same people who were accusing others of hype are just as guilty as hype now. I don't know. I'm wondering, in 10 or 20 years, are stem cells still going to be this wonderfully promising technology that hasn't quite gotten there? I mean, you think about gene therapy. I mean, 10, 15 years ago, that was going to be the next big thing, and now it's just basically dead in the water in terms of actual trials.
Right. Well, I think, there are two different questions here, and I think we should separate them. I mean, the first question is, who were these people who were screaming, it's all hype, embryonic stem cells are not going to work, and we shouldn't do it? And basically that came from a group of opponents, who disliked the research on religious grounds.
Right, that's true.
But they realized that they couldn't use their religious basis for being against the research to convince the majority of people to be against it. And so they had to pretend that there was some scientific reason. I mean, this is exactly like the creationism going into intelligent design debate. I mean, it's completely parallel, right? The creationists couldn't convince enough Americans to be on their side, and so they changed the name of the game. They make it intelligent design, and you are quite familiar with the tactic. And so here the tactic is, again, to pretend this has nothing to do with religion and pretend it's all just a question of science. So it's exactly parallel. They probably learned what to do from the intelligent design people. So that's the first issue. The second issue is this really hype or not? And I think that's a very complicated question, because embryonic stem cells potentially can be used for dozens, if not hundreds, of different human elements. And in some cases it's going to work, and in some cases it's not going to work. Mm-hmm. And if you're a bettor or you're a venture capitalist, you try to put your money in one or another. I think we're going to have cures. I mean, in a couple of years, is it going to cure everything that has been claimed could be cured? No, there's no question about that. But it's powerful technology, and I think it's actually more powerful, and even from the beginning you can see how it is more likely to work in some cases than gene therapy ever was.
So what makes it better?
Well, somatic cell gene therapy, the idea of putting...
I guess we should stop and say, just to make clear, gene therapy is where you take, somebody has a gene, a defective copy of a gene, and so it's causing them, say, cystic fibrosis or something like that. So So you stick the gene, say, into a virus, and you actually infect the person's cells with the virus, and theoretically, at least, the virus will insert this working gene into some cells. They'll make the proteins the person needs, and theoretically, they'll be cured. But it hasn't really been working out too well.
Yeah, and the problem has been the problem that has always been there, which is, one of the problems, is the efficiency by which you can actually put this new DNA, this therapeutic DNA, into a person's cells in a person's body. And the efficiency has always been outrageously low. I mean, I don't think they've ever gone over 1% of the cells actually taking up the DNA that you put into the patient. So right from the beginning, they had this huge problem, and they hoped they could solve it. But just theoretically, it's a very difficult problem of efficiency. With stem cells, the idea is that you can take these embryonic, so-called, I mean, they're not embryonic, right? They're iPS cells.
iPS cells, okay.
iPS cells, you take them from a patient, and especially when it comes to creating tissues or free-floating cells like hematopoietic stem cells.
Hematopoietic stem cells are the stem cells that are going to give rise to red blood cells?
That's right. So red blood cells to white blood cells to all the cells of the of the blood cell system. It seems pretty straightforward. People have almost reached the point. Scientists have almost reached the point where you can actually do that already, and so you can already imagine soon treatments for hematopoietic diseases of various kinds. And you can imagine the same thing for other kinds of diseases where all you have to do is get the cells to change their state. In other words, skin. So you start with a skin cell, you kick it back to the embryonic stage, and that's like being at the trunk of a tree, and now you're going to guide those embryonic cells up one of the branches to a particular outcome, particular kind of cell that the patient wants. So I can imagine some cases where that's going to be straightforward. Now, then you have cases like, okay, can we make a heart in the petri dish? I don't know if that's ever going to be possible. Or, you know, those are structured organs. They're much more difficult to deal with.
What about using stem cells in the brain? I guess that always boggles my mind that you've got this incredible network of hundreds of billions of neurons or whatever the number is, and the idea of just sticking in some cells, which will then, in a sense, kind of figure out where they are and what they should be turning into and so on, is pretty incredible.
Yes, and I since I am not a neurobiologist, I don't really have the expertise to decide whether it's how feasible it is to overcome things like Alzheimer's disease or Parkinson's disease. I mean, I've heard neuroscientists who are optimistic, but I don't know. I mean, it's amazing how quickly this technology is going forward. Whether that's really going to be possible someday, it's hard to actually put a probability on it right now.
Yeah. I mean, to sort of shift gears a little bit, I know you've been giving some talks about sort of the future of our species, really, and I'm sort of wondering, like, between stem cells and other kinds of technological interventions. I mean, what are your thoughts about where we might be going, or where we want to or don't want to go?
Technically the easiest intervention is also ethically the most contentious of all, which of course is to change the DNA within the embryo before the embryo develops into a child. And the genetic engineering at the embryo stage is something that's been practiced by mouse geneticists now for almost 30 years. They're very good at it. There are many, many generations beyond just adding or subtracting a gene. You can do very sophisticated changes to a mouse genome. And the technology has also been introduced into many other mammals as well, for agricultural animals like cows and sheep, for example. So there's no reason to believe that it couldn't work with human embryos. That's
so you change the genes in a fertilized egg so that the person, when they are developing and they grow up, their cells are making certain proteins that are desirable in one way or another.
Right, making some proteins and not making others. And it's not even clear that you'd actually have to add or subtract genes all the time. You might just want to turn genes up or down a little bit. I mean, Alzheimer's disease, for example, you might imagine reducing the predisposition or eliminating the predisposition to Alzheimer's disease just by a tiny little DNA change that just affects the production of proteins in a brain as a person gets older. And so you can imagine, I mean, already we know so much about genetic differences that provide people with resistance or cause susceptibility to a whole host of common diseases. And these are just coming out every month. We're learning this information. Very often the differences are just, one little piece, one little base of DNA rather than another. Mm-hmm. And so if you forget about ethics for a moment, theoretically you could take an embryo that a couple had, and you could put in all of these changes that would basically provide resistance to all common diseases. I don't know. I hear that sometimes, but I mean, another thing that is coming up all the time is that genes have lots of different effects. There's, I guess, people like to use the term pleiotropy, but it just is the sense that like genes do more than one thing, and so and there can be lots of trade-offs, and how do you find out what the trade-offs are in advance? I mean. Just to take an extreme example, I could say like, hey, guess what? I can make you resistant to malaria, but one of your kids might get sickle cell anemia. Sorry, how are we going to deal with that? I think that's the critical question, and in fact, there's an answer. And the answer, there's two parts to the question. One is that genetic networks are very, very complicated. No question about that. That's true. Diabetes is actually a complicated disease as well, and people can control diabetes with a single protein, insulin. Mm-hmm. So that's an example of a complex disease being overcome by one molecular component. A lot of these diseases, there are a lot of diseases which, yes, they're very complex inputs to the disease that you might be able to overcome by altering the expression of a single gene. But now the more important question. How do you know if there are any secondary unintended consequences? And I think the answer is that if you limit yourself to making changes that already exist in the human population, then you can do the experiment ahead of time. In other words, you can look at groups of people that have A at a particular DNA position or who have G at a particular DNA position, and you can study them and see. You can look at all the advantages and disadvantages of having one DNA letter or the other in a large population. Now, it doesn't guarantee you anything, but you actually can do the experiment without any manipulation ahead of time.
But you're also going to have to take into account sort of all the rest of the genes that the people have that are working together. There's this sort of genetic background in which these different versions of the gene are going to be working.
But I think that if you do a population study, you automatically take that into account.
Okay.
In other words, if you look at a diversified population and the only difference between your two groups is one genetic alternative, and everything else is random. You've kind of accounted for that, and in a sense, that's what all of these new studies of what are called whole genome association studies are doing. Now, there are many issues with genetic engineering of human embryos. I don't want to downplay them, but it just is in the case with embryonic stem cells. A lot of the arguments are easy to dismiss. There are arguments being made by people who are offended and frightened by this technology for reasons that have nothing to do with the science, for much more basic ideological or religious reasons.
Such as?
Well, such as human beings should not be playing God. It's God's right to decide the nature of each human being that is born, and we shouldn't be mucking around in the genome, changing what God's wishes are. I think that's the major objection. Often that objection is not consciously expressed. It is sort of a subliminal, subconscious notion that we should just leave nature's good. We should leave nature alone. And then people come up with all sorts of scientific reasons of why we shouldn't do this, why it's going to be horrible. But I think you can overcome some of them. Just like this example of, every gene interacts with thousands of other genes in very complex ways, which we'll never understand. That's true. But we don't have to use that approach if we already have the two different gene variants in the population, we can then look at the advantages and disadvantages of each, and you do a risk-benefit analysis.
So that actually sort of brings up, I guess, maybe the last thing we have time to talk about now. It's something that I was thinking about while I was reading Challenging Nature, your book, and you kind of brought it up now. I mean, in your book, you really do this powerful job of taking on these arguments about these arguments against stem cells or against genetically modified foods and so on, based on premises that you don't think are right, and and that just don't make sense. And in this, I'm just sort of wondering, if you set aside these, we'll call them ideologies, just not in a pejorative way, but just these ideologies about what's good and what's bad. There's the idea, sort of a very, you could call it sort of a very religious ideology, in the sense that argues that even a single embryonic cell is an individual human in the full sense of the word. Okay,
right.
Let's set that aside. There are, the opponents of genetically modified food sort of talk about the integrity of nature and the integrity of species, and you shouldn't tamper with nature. Set that aside. But you actually didn't talk much in the book about what you think are good kind of ethical guideposts, or just sort of a framework for thinking about what we should do and shouldn't do. I mean, how do we make these sorts of decisions if if we set those other things aside, well, how do you think we should be making decisions? Is it all risk-benefit? I mean, and how do you decide what's a risk and what's a benefit?
Besides being a scientist, I would say if you had to pin an ideology on me, I would call myself a secular humanist. This is a very difficult question. I think that risk-benefit is a scientific, technological approach. And if you're talking about outcomes being positive or negative, you can evaluate risk-benefit in terms, in physical and physiological terms, and in tangible terms. You can determine whether a particular technology has benefits that outweigh risks or not. When it comes to culture and religion, then different cultures and religions are going to have different values that they express. And I have to tell you that it's not that I'm opposed to people expressing those values. I had a wonderful time talking with the bishops at the American Council of Catholic Bishops, and I was delighted to talk to them because they told me what they thought. They told me what they believed, what their values were, where those values came from, and I felt that they were being honest to themselves, and they were honest to me in telling me what they thought. So I didn't have any problem with that, even though I don't agree with it. I have more of a problem with people who are making up scientific objections to something based on a hidden religious or cultural agenda, which may even be hidden to them. So, but to answer your question, what should we care about? Of course, that's a very difficult question. I think that we should care about helping human society, humankind. And it's very, very difficult. I mean, this is what philosophers and ethicists debate all the time. But it should be done I believe, based on rational analysis of what is positive or negative for people.
Well, with the time has flown. I mean, there were actually a bunch of other things I wanted to talk about, but let's maybe we can try and talk about them another time.
That would be great. Be happy to do that.
All right, so let's sign off now, and we'll talk again soon.
Okay, Carl. Take care.
Take care.